So it's a pleasure to welcome back today's speaker, Parthasarathy, to Berkeley. Some of you may remember, those of you who are sort of long in the tooth may remember that he was an undergraduate here in the 90s. He was the department citation winner in 1997. I had the good fortune to teach him in quantum mechanics 137A in the spring of 1995. I know that because I found his blue book for his final exam. He did very well. I'd like to, I found it during the pandemic because I'm a hoarder, but for the purposes of this introduction, it's because he was such an outstanding student. He did get an A+. After getting his undergraduate degree here, he went to University of Chicago, where he worked with Heinrich Jaeger and Tom Rosenbaum. And then he came back to Berkeley and worked with Jay Groves for his postdoc as a Miller postdoctoral fellow. And since then, he's been at University of Oregon, where he is now the Alec and Kay Keith Professor of Physics. Raghu is a very distinguished scientist, as you'll hear. He's, among his many honors, he's a fellow of the American Physical Society, and the citation is for his creative and innovative contributions to biological physics, some of which you'll hear about today. One of the things I really admire about Raghu is his dedication to physics education. That started when he was an undergraduate here. He co-developed a course as an undergraduate about building a radio telescope. Since then, he has this great blog called The 18th Elephant about physics and biology and education and art. Very interesting. And he's also written a nice book called So Simple a Beginning, which is a quote from Darwin, The 4 Physical Principles That Shape Our Living World, which I think is now in multiple languages. So that's by way of introduction. Please join me in welcoming Raghu back to Berkeley, and he'll tell us about gut microbiomes and their physical concerns.
Yeah, this one on? Good. So thanks a lot, Dan, for the introduction. That was really nice, and thanks everybody for the invitation. Yeah, as Dan mentioned, I've been here both as a postdoc and as an undergrad, and as an undergrad as a physics major. So I spent a lot of time in this room attending colloquia and stuff like that. So it's really quite thrilling to be here again and on the other side of the podium here. So I'm going to tell you about some of the things that my group has been doing, looking at gut microbes and looking in the quite literal sense. So these days, pretty much everybody knows that each of us carry along with us trillions and trillions of microbes. And most of these are resident in our gut, and we have these gut bacteria that outnumber actually the human cells in our body. And this gut microbiome does a lot of things for us. It plays roles in processes like digestion, training of the immune system, the early development of organs. They play roles in various diseases, especially rather complex diseases, things like diabetes and malnutrition and stuff like that. So we're part of this gigantic ecosystem that each of us carry along with us. And you might be wondering, well, all that's well and good, but why might a physicist care about this? So to answer this question, let's take a step back and think about basically any macroscopic ecosystem. So if you think about the Serengeti savanna, or you think about tide pools on the coast, it would be very hard to make sense of how those ecosystems work if you didn't know anything about their spatial organization and dynamics. Like if you didn't know that wildebeest travel in herds and grasses don't run around, okay? Or that you didn't know that starfish move around and hunt and the tide comes in and out twice a day. It would be very hard to understand and develop a deep understanding of how those systems work. So given that, you might wonder, okay, what is the spatial organization and structure of these gut microbial communities that are in all of us? Are bacteria motile? Are they aggregated? Where are they in the gut? Knowing these things, we might be able to get a good sense of how species can coexist, how competition might occur, how we might have resistance or susceptibility to invasion, and so on. At an even deeper level, we might want to know not just how are things spatially organized, but what are the factors that influence spatial organization and dynamics in gut bacterial systems. So we actually don't know very much at all about this, basically because these kinds of questions are very difficult to assess with standard techniques. And by standard techniques, I mean nearly everything we know about the gut microbiome comes from doing things like taking fecal samples and doing sequencing of like DNA or RNA to figure out what's there. So this tells us what species are present. It tells us what genes are present, what genes might be being expressed. It is immensely powerful, and it's through things like this that we've figured out a lot of the things that I put on the first slide about connections to disease and physiology and so on. But as you might imagine, taking fecal samples, it has hardly any temporal resolution and really no spatial resolution. You're not going to answer these biophysical questions from that. So what can we do? Well, starting about a dozen years ago or so, we thought, okay, let's tackle this by taking an approach that's been very powerful throughout all of sciences, but I think especially powerful in physics, of either finding or constructing a good model system and then observing it. So our model system is the zebrafish. And these are very popular as a model organism for some very well-known reasons. They're vertebrates, so they're physiologically similar to humans and other vertebrates. They're genetically amenable to a lot of interesting techniques. And also when they're young, when they're larvae, which is what I'll be talking about, they're quite transparent, so you can look into them. In addition, it turns out that you can prepare zebrafish to be initially germ-free. That is, devoid of any gut microbes or any microbes at all. And then you can take those germ-free zebrafish and just introduce into their surroundings particular bacterial species, including ones that you've engineered to have fluorescent probes, for example. So you can do controlled experiments in terms of species or timing or things like that. And especially a lot of these techniques for developing germ-free or so-called notobiotic zebrafish have been developed actually by our colleague down the hall, Karen Gilliman, also at the University of Oregon, with whom we collaborate on quite a few of these experiments. So shown here is a little just cartoon of a larval zebrafish, and the gut here is outlined in magenta. So I mentioned that the larval zebrafish are pretty transparent, but it's still kind of non-trivial to image gut bacteria. And that's because we need to image quite large fields of view. So the gut is like 1 millimeter long, even in larvae, and then hundreds of microns by hundreds of microns. And we want single bacterium resolution. And a bacterium is about 1 micron. So we want large fields of view. We need to image in 3D because we expect structure to be three-dimensional. We need to image for many hours without inducing a lot of photo damage or phototoxicity. And we need to do this three-dimensional, gentle imaging very fast because roughly every minute or so, the gut is doing this. These peristaltic contractions that are going to come up actually throughout this talk, these peristaltic contractions that your guts are doing as well, especially if you just had your cookies or whatever upstairs, of transporting stuff along the gut. So to do all this, or the technique that makes it possible to image inside the zebrafish gut and with all of these characteristics, is light sheet fluorescence microscopy. And here, many people might be familiar with this because of the kind of amazing work from Eric Butzig and colleagues. Light sheet microscopy is a technique that you can embellish in lots of different ways, including structural illumination, things like that. But the basic principle of it is something very, very simple. We're doing fluorescence microscopy, so we're shining light of one color and then looking at emission of another color. But we're shaping that excitation light into a thin sheet. And that sheet intersects our specimen, like a larval zebrafish, just indicated by this golden orb here. Then we're imaging the emitted light from the perpendicular dimension, like through this lens over here. So at any instant in time, we're getting a 2D optical slice, and then we can construct a 3D image just by scanning in that one remaining dimension. So it's very fast, very efficient, and it makes this sort of imaging possible. So these days, you can actually buy from several different manufacturers light sheet fluorescence microscopes. We build our own, and we built a couple of setups, one of which, the kind of key region of which is shown here. So here is this lens, the one that's magnifying down that excitation sheet. Here's the perpendicular lens. And you see here a bunch of glass capillaries. And at the end of each glass capillary is a plug of agar gel. And in each plug of agar gel is a larval zebrafish. So they're not free swimming, but they're alive, and they're able to, as far as we can tell, they're happy sitting there in their agar gel. So what do we see then when we look at their gut microbes? So I'm showing you here, oh good, it is playing, the gut of a larval zebrafish that started out germ-free, and then was colonized by just one zebrafish native gut bacterial species, some of which are expressing a red and some of which are expressing a green fluorescent protein. And in this movie, we're scanning through in depth. And what you can notice are a few things. You can see like little individual red and green speckles, and those are individual bacteria. And you can also see dense three-dimensional aggregates, these blobs of red and green. So from the very beginning, this was actually both surprising and reassuring. Surprising because we actually had no idea what to see. So before we had done this, nobody had ever done any bacteria-resolved imaging of gut microbes in a live organism. So we had no idea what to expect, and we could have been extremely unlucky. It could have been that just, the gut microbiome is just a homogeneous soup of bacteria. But it turns out there is structure. We see, for example, this kind of aggregation. And moreover, there's aggregation of different sorts that different species can do. But nearly every species that we look at is forming some kind of three-dimensional structure, some kind of dense aggregate. I'll show you an exception to this in a moment. We see this kind of aggregation in nearly everything we look at. And then more importantly, it turns out that this kind of structure that we observe among the microbes ends up being a major factor in nearly every kind of physical or physiological or whatever you want to call it process that we've looked at. So I'm going to tell you a couple of stories about the kinds of things that we find by imaging here. So the first story I'm going to tell you is one that started several years ago, although it actually leads into some ongoing work. And I'm telling this one to start off with, both because I really like it, but also it's kind of neat that the key driver of this one is Brandon Schloman, who's in the audience there, who's a postdoc in Hernan Garcia's group, who hopefully many of you have had a chance to interact with in the last couple of years. So this first story, oh, and I should also point out, actually maybe I'll even pause. Feel free to interrupt with questions. Don't feel like you have to wait until the end. And I'll even prompt you any questions about the methodology now. Yeah, Dan.
So the red and the green bacteria are the same species?
In this example, correct.
And
Yeah.
so when you see a clump of green, it's because the green bacteria has divided?
Correct. Yeah, so that's a good observation. So even here, you can tell there's interesting things going on because it's not a salt and pepper of red and green, but actually like clonal colonies of red and green. Yes. Yeah. So even from this image, you can tell that there's like rapid growth going on. Oscar, yeah.
Where was this gut colonized by just two cells initially, or?
Right. So that you can't tell from this image, but we've done things. So the short answer is the initial colonization appears to be very small numbers of bacteria. So we can image, soon after we introduce bacteria into the water, and we'll see like 8 bacteria in the entire gut. So it looks like the colonization is a rare event. Once the bacteria get in, they start multiplying like that. Yeah, that's a great question. Yeah?
And these are not scanned through, these are just set. It's not like you're progressing the...
That sheet, so we're actually scanning the fish through that sheet. Yeah, so, but that's very quick. We can do that in less than 30 seconds or so.
So is the temporal thing that we're seeing there, is that because of the scan or because of the change in the bio?
Oh, it's because of the scan, yeah. So we're looking at a 3D, so keep in mind the image is basically a 3D image, and to show this on a 2D screen, I'm scanning that sheet.
Exactly, but it's not moving in time as well.
Not on this time scale, right, yeah. You'll see later on, I'll show movies of what happens to this over hours, so we will see things where it's changing, but not in this one.
So when you said at the end of the filament, there's a what? I couldn't hear you.
Oh, like the fish is held in a blob of gel.
A blob of gel. Okay,
Yeah.
I didn't...
No, great. Excellent. Anything else? Okay. So this first story has to do with antibiotics. So it's been known for actually well over a decade that antibiotics induce large changes to the composition of the gut microbiome in humans as well as other animals. And that in itself is not at all surprising. I mean, they're antibiotics. That's what antibiotics should be doing. The surprising part is that antibiotics can induce large changes in the microbiome, even at quite low concentrations, well below the so-called minimum inhibitory concentrations. And this is interesting, but it's also important because low levels of antibiotics are often found as environmental contaminants. So how is this happening? Why might low-level antibiotics have an impact on the gut microbiome? So we decided to look at this, and we took a very kind of reductionist approach of taking like a model antibiotic. What I'll show you is ciprofloxacin, which is a commonly used antibiotic, and it's also a common environmental contaminant. And we're going to look at what this does just a single species of gut microbes. So we have a germ-free fish that's colonized by a single bacterial species. The first species I'm going to show you is this guy. So this is a zebrafish native Vibrio species. And here what we're looking at is just one optical plane in real time. Okay? So we're looking at one slice in real time. Each little speck there is an individual Vibrio bacterium. And this species is our least aggregated species. There's a tiny bit of aggregation, but the vast majority of these guys are just zipping around. They're very motile, planktonic, individualistic. They're just like a swarm of bees there in the gut. I also just really like this movie, and I always tell people this is what you should imagine is going on in your guts as well. if that's reassuring or not. But anyway, our swarm of bees there. So what happens if we add weak antibiotics to this one? So we have these bacteria, or we have zebrafish colonized with this Vibrio bacteria. We add in a low level of antibiotics, about 1/10 of the so-called minimum inhibitory concentration. We measure the density of bacteria in the water, and it's somewhat lower, but about 10 times lower than it would be with no antibiotic. Then we can take the gut, dissect it out, just determine the overall population of bacteria, and it's not 10 times lower, it's 100 times lower. Okay? As we kind of expected from these sequencing-based studies, the weak antibiotics have a big effect. So what's going on? So let's image. And so this movie is, again, a single plane in real time. But now, what you can hopefully see is those bacteria are now not so zippy. What used to be a swarm of bees, now they're much less motile. It looks like a bunch of slugs. I actually gave a talk at Santa Cruz, and I learned what a bunch of slugs is. Does anybody know? It's a cornucopia of slugs. So that can be your takeaway message for the day. So our swarm of bees has turned into a cornucopia of slugs. They're much less motile. And if you look carefully, it may be hard to see from out there, but they're also they're longer. They're filamented as well. So this has an interesting consequence. Now if we look over several hours, it's the lower movie here, what we find are those filamented, less motile bacteria are basically pushed by these peristaltic contractions of the gut and are transported down the gut and expelled from the fish. And excuse me, these bacteria are alive. We can do like a live dead stain and show that the expelled bacteria are mostly alive, but they're no longer able to persist in the gut. So this enormous population drop is not due to death, but rather to not being able to withstand the physical environment of the intestine. Now, if we were really clever, maybe we would have guessed that, because this filamentation and loss of motility are actually quite common stress responses of bacteria to antibiotics. And so what's going on is that that process is occurring, and then those effects are essentially amplified or kind of more consequential because of this coupling between intestinal transport and aggregation. So the intestinal transport basically pushing aggregates along. So that is what happens to a very motile species. What if we instead do the opposite, or look at the other extreme, a bacterial species that is very aggregated? So here what I'm showing you is, again, a gut monoassociated just with one gut bacterial species. This species, it's an Enterobacter species, is our most aggregated species. About 99% of the population exists in dense aggregates. They might be very large aggregates, like that one, or medium-sized aggregates, or smaller aggregates, but nearly everything is aggregated. So what will happen if we add weak antibiotics to these guys? So this is the interactive part of the talk. So same deal. We're going to add weak antibiotics. What's going to happen? The population will go up. We'll see not much of a change at all. We'll see a similar effect as with that motile Vibrio species, like a 100-fold drop in population in the gut, or we'll see a larger population drop than Vibrio. We don't have a way to do this totally anonymously, but I'll just ask you to raise your hands. How many votes are there for A? B? C, D. Interesting. It's B and D seem the most popular. I would have guessed B because they're already aggregated. What are they going to care if they aggregate some more? And the answer turns out to be D. We see, so congratulations to those of you to D. But like I said, I would have guessed B. But it turns out it's an even bigger effect than we saw with the Vibrio, that very motile species, about a 1,000-fold drop in the population compared to not having antibiotics at all. So what's going on? Well, again, if we image, we see something quite dramatic. The upper image, we see Enterobacter without antibiotics, and this is the same image I showed you before, clusters of all sizes. With antibiotics, sometimes we don't see any bacteria at all, but if we do, they're in the very large clusters are there, and the small clusters are absent. We see far fewer small clusters with the antibiotic than otherwise. So we see predominantly the large clusters, and as I mentioned, large aggregates are very susceptible to transport, and those are transported out of the gut, and so we end up with this enormous population drop. So This led us actually to a very general question. Oh, yes, yeah, great. Yes, if you didn't hear that, again, they're predominantly alive when they come out, right? So we're not killing the bacteria. So this led us actually to a very general question, which is, okay, so let's suppose you're an aggregating bacterial species. And as we've seen, aggregation is coupled to transport. So if you're aggregating, the aggregates are going to be transported out. How can you maintain a stable population in the gut? Well, it must be that aggregating species, like the nucleation and growth of new aggregates, is really crucial. So you're, as actually you can see in this movie of Enterobacter over a couple of hours, the large aggregates are transported out, but there's plenty of new aggregates nucleating and growing to take their place. So can we turn that kind of cartoon picture into something more quantitative? Can we come up with a kind of biophysical model of cluster dynamics? And here I'm going to tell you the kind of very quick and rough version of the story. If you want to know more of the details, check out this paper there, which has a lot of neat stuff in it. So by staring at these movies a lot, we in this case is Brandon, realized that there's really kind of 4 underlying processes. And I do want to highlight that it's really just staring at these movies that makes things possible. There's no kind of pre-existing theory there to really guide us. So we realized there's kind of 4 key things going on. One of them is just growth, as actually came up already, just cell division. So we have, cells dividing, so there's some rate at which we'll go from a cluster of n to a cluster of n+1, just from division. If you didn't have any kind of constraints, you would just have exponential growth, so your growth rate of your number of cells in a colony is just proportional to how many things are there. If there are constraints, like a finite carrying capacity, like this K here, you'd expect logistic growth for anything growing there. So we have just growth and cell division is one process. Second, we have aggregation. So we can see aggregates coming together, basically being pushed together, again, by peristaltic motion and transport. And the larger a cluster is, the more likely it is to aggregate. Basically, you have more area there for aggregation, so you have some kind of exponent there for some kind of a kinetic rate that's describing aggregation. There's fragmentation. So you might imagine that a cluster could break up into smaller clusters. Interestingly, we never see a cluster, for example, fall in half. Rather, what we see are individual bacteria coming off of clusters. So fragmentation in the sense of like individual bacteria leaving clusters, which is probably actually related to cell growth, but we're kind of describing it as a different kind of process there. And finally, this expulsion I mentioned, just being transported out of the gut completely. So for all of these, you can just write down little kinetic equations for them and do either simulations or some analytic theory to say, okay, if you have all of these processes going on, what should you expect in terms of steady-state populations or other things that I'll mention in just a moment. You can see, again, the paper for more of the details there. So you can put together that kind of model, but one thing that's very nice about it is that we can actually say things about many of the parameters in each of these steps from the experimental data itself. So we can actually directly measure things like the growth rate in the gut, and actually even just cluster-specific growth rates there. And for some of the other parameters, we can't directly get them, but we can get things like ratios of various parameters, again, from the observations. And if we do that, there's a lot of things one can get out of that. I'm going to show one kind of simple way of looking at that, which is to make kind of a phase diagram where I put here like the ratio of like growth rates and expulsion rates on one axis, and the ratio of fragmentation to aggregation on another axis. And in the color here indicates what this steady state population should be. So dark blue is basically extinction, and all of these lighter colors are some finite number of bacteria that we should expect in the gut. So if I take that Enterobacter species and ask what are its parameters without antibiotics, I get kind of an expected population around here, which is nicely in the non-zero regime. Oh, there it is, yeah. Then, with antibiotics, what we have is then a reduction in the growth rate and, importantly, a reduction in that fragmentation rate, or the relative ratio of fragmentation and aggregation, which pushes these guys across this boundary into the regime where we shouldn't expect really a steady-state population consistent with what we observe. You may be wondering, what about the Vibrio? Without antibiotics, it's basically not aggregated at all, so it's kind of off scale. But then with antibiotics, it gets pushed into a much lower abundance, but not actually the complete extinction regime there. There's more you can get out of this. You can actually ask, what is the distribution of cluster sizes you should expect? And that's actually kind of the more sophisticated version of this model that you can read about there. But you can measure that and also compare like the model predictions to the experiment as such. So like I mentioned, this leads us actually in a bunch of interesting directions. One, which we've started on, is what happens now if you have more than one species? Are there interesting consequences for things like competition between bacteria due to things like differential interactions with antibiotics? Another one, which we've thought about for a while but have not actually done anything with until being spurred to now, is can this be related to phenomena such as antibiotic resistance? And this is especially exciting because actually Oskar Halacek and I now have a new kind of collaborative project to look at exactly that. So you can ask things like, are there consequences of like aggregation or expulsion or things like that to either the development of resistance or determining like which species develop resistance and which don't, or which kind of subpopulations develop resistance and which don't. Finally, this, maybe I'll comment on at the very end. You can, in some sense, think of the antibiotics in that Vibrio case at the beginning as inducing, in a way, a transition between a motile and an aggregated state. Are there perturbations that might do the opposite? And we think we've actually found one, but you can ask about that at the end. So we've seen in that first part of things consequences of this coupling between aggregation and transport. So I'm going to tell you a different story in a moment, but maybe before that, any initial questions about that point? Yeah?
Two questions. One, the evolution of these microbes moving from the parameters, like over human or animal relevant timescales?
Oh, you mean evolution? Okay. So the question, if you didn't hear it, was, yeah, could evolution tune basically these parameters? Right. So that's a great question. In a broad sense, definitely. So first of all, I should note that these species that I've been telling you about are ones that were isolated from the zebrafish gut. And if you just take some random species like everybody's lab favorite E. coli, it's a terrible colonizer. So in some sense, these have already evolved to be good colonizers and to exist there. So that's part of the answer. The second thing, which might be more of what you're thinking about, could you, on like kind of experimental timescales, do sort of experiments involving selection? Could you further evolve these? And I think the answer to that, we haven't done anything with that, so I'm kind of speculating, but I think the answer to that will be yes, because there are some other experiments we've been doing, and especially our collaborators have been doing on like kind of directed evolution. Can you induce like in vitro aggregation, like changes in aggregation state, stuff like that? And interestingly, in just 30 passages or something like that, you can make species aggregate or disaggregate. And I think you could construct an experiment where that's going on in vivo as well. So I think the answer to that is yes, but we haven't really looked at it. It's a very good question. And that's actually very relevant to like antibiotic resistance and that kind of thing also. Yeah, anyway.
I don't know if this is a better question for the end. But I guess that you motivated some of this by saying how there were antibiotics will like wipe out colonies in our gut.
Right.
Showing how. So when people or organisms, whatever, people are not, when they like, let's say you do wipe out
Right.
the homeostasis.
Mhm.
You said E. coli was really a bad colonizer.
Correct, yeah.
Like, is this pointing to ways that one would try and properly colonize?
Yeah, that's a great question. So I hope so, but we really don't understand well at all what makes a good colonizer. And that's not just true of our work, but just in general, there's, as you know, a huge interest in like probiotics and stuff like that. And one thing people actually know is that a lot of like, ingested probiotics, they don't colonize at all. Like it's actually just it's a total crapshoot of what actually sticks in the gut and what doesn't. What, so to speak, exactly. That wasn't planned. So what makes something a good colonizer is, I think, a really key question, and it's very poorly understood. There are I'll comment a little bit. We know a little bit about that for some of these bacteria, so I might comment a bit more about that at the end too. Yeah, Dan.
Is any of the aggregation preserved in the fecal output?
So the output is also aggregated.
Did you look at human
Oh.
aggregation that way?
Yes, so Brandon and I used to talk about this a lot. I think there's a lot, there have been just a handful of studies, some of them actually really old, like decades old, noting the existence of like aggregates in fecal samples, experiments, I'm glad we don't do, but noting the existence of that. And I actually feel like there's a lot of information in like cluster sizes and things that people have not looked at. So I suspect that you could use aggregation state kind of as a measure, a proxy measure of things that might be going on in the gut, and I think that's very underexplored. Yeah, Jay, or not Jay, sorry, just looking at you. Yeah.
Do biofilms play a role in aggregation of bacteria?
Yeah, that's an excellent question. So this depends a lot how you define biofilms. So interestingly, at least in zebrafish at these ages, we never see biofilms kind of coating the surface. The aggregates we see are these dense 3D things. We've done a little bit of stuff on the expelled aggregates, and there are, kind of polysaccharides and things that are commonly found in biofilms that are there. I think it's an open question whether these are really similar to like in vitro biofilms or not. So one thing, whenever I talk to biofilm people, it seems kind of contentious whether to call these non-surface attached things biofilms or not. But I think there are similarities, but I'll leave it at that. Cool. Okay. So in what we've seen so far, we've been looking at, or one of the things we've seen are couplings between aggregation and intestinal transport. So what about the opposite? Can microbes alter basically the physical environment of the gut? So can gut microbes alter intestinal mechanics? And this is a story that actually started with some observations several years ago, but is continuing to the present with a paper actually that just came out a couple of weeks ago on a partial answer to this mystery. So I mentioned actually just a moment ago that nearly everything we do looks at zebrafish native gut microbes. But one exception is a few years ago, we got interested in Vibrio cholera. So that is the bacterium that causes the disease cholera, which still remarkably kills quite a few people per year. Cholera has been intensely studied for well over a century, but there's still actually some very basic mysteries about how it works, including how does it invade an already densely colonized human gut? So we, especially a graduate student, Savannah Logan, and some collaborators, especially Brian Hammer at Georgia Tech, who's been studying Vibrio cholera for a long time, got interested in something that Vibrio cholera has called the type 6 secretion system. Actually, quite a few bacteria have this. And it's this syringe-like stabbing apparatus with which Vibrio and other bacteria puncture adjacent cells and insert toxins. So we wondered, what is this type 6 secretion system doing in vivo? So we got some Vibrio cholera, exposed the fish to it, and what we found is the following. So here, I'm showing you a movie of just the gut tissue itself, these peristaltic gut contractions. And this is a zebrafish colonized with Vibrio cholera that has a defective type 6 secretion system. This movie looks exactly like it would if the fish weren't colonized with any bacteria at all, or were colonized with pretty much every other like normal bacteria. We see these nice kind of propagating contractions in the gut. There one goes.
Is that flicking stuff along with that blood?
Yes, yeah. Blood cells zipping around. Here is a zebrafish colonized with Vibrio cholera that has a type 6 secretion system that's always being expressed, so it's always stabbing. And as you can hopefully see, even just by eye, the contractions in the lower movie are intense, much stronger than in the upper movie. So this was a big surprise, actually. We were not expecting this, and nobody had seen this before. So we can take these movies and do like image velocimetry and quantify like the frequency and amplitude of these propagating gut contractions. And what we find is, if you put numbers on it as well, with this type 6 secretion system, the amplitude of these gut contractions is about 100% larger than without the type 6 secretion system or without bacteria at all, with actually a large variance also. So this type 6 secretion system, there it is. And at its tip, it has something, in Vibrio, it has something called an actin cross-linking domain. And if you know what actin is, or actually, if you don't know what actin is, it's this cytoskeletal protein that is found in eukaryotes, so like you and zebrafish and stuff like that. So this actin cross-linking domain is acting on eukaryotic cells like the zebrafish cells. If you remove that actin cross-linking domain from an otherwise functional type 6 secretion system, so it actually can still kill bacteria, but not eukaryotic cells, those gut contractions go back down to normal. So deleting the actin cross-linking domain is your normal gut contraction. So for years now, we've been wondering, what could possibly be the connection between bacterial stabbing and intestinal contractions? And Julia Ngo, who was a recently graduated grad student in the group, decided to tackle this by using not Vibrio cholera, but a zebrafish native Vibrio, which colonizes to high abundance because it's zebrafish native. It lacks the cholera toxin, so interpretation of things is simpler. And she made a Vibrio lacking this actin cross-linking domain and did a lot with it. This Vibrio species is the same one that I showed you before, this very motile, highly colonizing species there. So just like Vibrio cholera, if you have just the wild type Vibrio, you have strong contractions. If you take out the actin cross-linking domain, you have weaker contractions. And even the magnitude is quite similar to what we had before. So with the actin cross-linking domain, you have about 100% increase in the strength, the amplitude of these gut contractions. Interestingly, for all the physics students in the room, I mentioned amplitude, but what about frequency? Frequency does nothing interesting. Same frequency of contractions in all of these things. So, right, so this Vibrio behaves in a nice way, and now using this, what's the connection between bacterial stabbing and gut contraction? So for various reasons, we thought the immune system might have something to do with this. And this is in part because, and specifically macrophages, which are one of the cells of the innate immune system. So in part because for unrelated reasons, we had been looking at these Vibrios and their kind of immune stimulatory response. So it turns out they really induce a very strong immune response. Here in magenta, we have the Vibrio bacteria, and in green, we have a fluorescent reporter of something called the TNF-alpha pathway. It's basically like a marker of immune stimulation. So you see a lot of green cells there, many of which are macrophages. In addition, other groups in recent years have shown that these macrophages, these innate immune cells, communicate with the so-called enteric neurons, so the neurons that are lining the gut that are influencing like muscle and contraction and that kind of thing. So people have shown that macrophages and these enteric neurons are in close proximity to each other, and have even established some of the kind of signaling pathways between macrophages and enteric neurons, both in mice and actually in zebrafish as well, where people have shown that if you deplete the number of macrophages, stuff like food gets transported through the gut faster in these animals. So we suspected macrophages, and what Julia did is basically create fish that have fewer macrophages. You can do these thanks to these beautiful techniques that exist now of using like CRISPR-based methods to interfere, for example, with transcription factors that are involved in the differentiation, the formation of macrophages. So here we mutated this IRF8 gene. And if you do that, you don't completely get rid of macrophages, but you have about half the number of macrophages that you do in a normal fish. Here's a fluorescence image of macrophages. There's far fewer if we do this IRF8 mutation. So we can make fish then that have far fewer macrophages, fewer immune cells. What do their gut contractions look like? Well, if we deplete the macrophages, if we have fewer macrophages, we have gut contractions that are about 100% stronger than they are in normal fish. And again, with no change in frequency. What's more, if we take those macrophage-depleted fish and add Vibrio to them, it doesn't matter if we have the actin cross-linking domain or not, the gut contractions are pretty much the same, suggesting that the Vibrio effect is acting through these macrophages in the system. So doing these experiments, Julia made a very interesting observation, which is there's something else going on in these fish. If we look at the vent, that is like the anus, the posterior end, there's a lot of indicators of tissue damage. We see a much wider vent there, much wider with this wild-type Vibrio than if we get rid of this actin cross-linking domain. And there's a lot of like just ugly-looking rounded cells, which are indicative of cell death. And if you do a specific stain for dead cells, you find a lot of dead cells. So we thought, OK, there's a lot of tissue damage. There's a lot of cell death. Maybe what's going on is that the Vibrio bacteria are killing the macrophages. And so that's like not having very many macrophages, which gives you strong gut contractions. So that's the mechanism of what's going on. So if that's true, we should see a lot of dead macrophages. And Julia looked, and there are dead cells in cyan. There are macrophages in magenta, but we don't actually see a lot of dead macrophages. And what's more, it doesn't matter if we do or don't have this active cross-linking domain. The fraction of macrophages that are dead is small and unchanged regardless of that. So it looks like, we do not find a large actin cross-linking domain dependent fraction of dead macrophages. So what's going on? Well, recurring theme, we decided to just look more. So here, what I'm showing you is just a quarter of a full scan of the zebrafish gut. In cyan is that reporter I mentioned of immune stimulation. There's a lot of stimulated cells there. And in magenta are macrophages here. You can actually see these just kind of nicely outlining the tube of the gut there. There's a lot of other just beautiful stuff going on in this one. And what we can do is take these images of the entire gut, again, for hours. So if we do that, good, what we find is that these macrophages, they're very, very motile. And they preferentially are accumulating at that posterior vent where that tissue damage is occurring. So we get, after these two images are about 10 hours apart. After 10 hours, we get a lot of these macrophages near the vent, and a scarcity of macrophages kind of along the midgut, lining the midgut there, where the neurons are. And the cyan here are the neurons. We track all of these cells so we can quantify this. So here I'm showing just the number of macrophages as a function of position along the gut. And as time goes by, if you look at these two spots especially, we see fewer cells here, more cells here as they're migrating from the midgut to the end. And you can quantify that as the difference between these two locations. And again, you see this big migration of cells. And overall, the number of cells is actually pretty much unchanged. So putting all of this together, what we've got is the following. We have this actin cross-linking domains, this Vibrio type 6 secretion system with its actin cross-linking domain is inducing strong gut contractions. Similar, very similar, in fact almost identical in magnitude to what we see if we get rid of macrophages genetically. The Vibrio is causing this posterior tissue damage. It's not disproportionately killing the macrophages. It is activating them, so we have this TNF-alpha response. And it's inducing this redistribution of macrophages from the midgut to the posterior, leaving fewer macrophages near those enteric neurons that are lining the midgut there. So this isn't a proof, but it seems like a very likely and plausible mechanism that it is this macrophage redistribution that's the mechanism for these Vibrio-induced gut motility changes. As I mentioned, it's not a direct proof because it would be nice to just directly alter the macrophage enteric neuron communication, but at least so far we don't know how to do that. But all the signs are pointing in this direction, and it's giving us at least a clue of what this quite dramatic phenomenon might be due to, which is much more than we've had for the past several years. The other thing that I really like about it from kind of a biophysical point of view is that, this mechanism for altering gut motility doesn't actually require any new biomolecular signaling pathways. We're not changing what proteins are, or at least in this picture, we're not changing what proteins are secreted or things like that. Rather, it's changing the spatial organization of the cellular players there, and that's what's altering the overall physiology of the host. So also, it's kind of neat that this illuminates some very old observations from our group. So does this kind of thing matter for communities of gut microbes? So we've known for quite a while that this Vibrio species, here indicated in cyan, is a really good invader. So if you already have bacterial species present in the gut, Vibrio is really good at just coming in, growing to high abundance, and sometimes even kicking out the original species. And here, Vibrio is in cyan, and Aeromonas, that species back from the very first movie, the red and green one, is indicated in magenta. And as you can see, these aggregates of Aeromonas are essentially being expelled from the gut due to very strong, what we now realize are very strong gut contractions. So we think this not only matters for kind of overall what are the dynamics of the host, but actually for gut bacterial communities. So I'm going to skip this movie. So in that whole picture, what we have going on is this migration of macrophages being kind of key to things. And interestingly, in the last couple of years, we've been looking more at immune system dynamics and that kind of thing. And macrophage migration is coming up actually in other kind of bacterially mediated contexts as well. So just to briefly give you an example before ending this, here's something else that, here's something else kind of related. So I showed you that those macrophages are moving to that posterior vent where we have all that tissue damage going on. So that in general is what macrophages do. They'll go to sites of tissue damage, kind of driven by signals emanating from that location. You can also see that if you do something like cut the fin of a zebrafish. Zebrafish are actually a very popular system for looking at wound healing. Even adults will actually regenerate their fins completely if you cut them, which is quite remarkable. So here, I'm showing you in cyan, neutrophils, and other immune cell. You can ignore those. And in magenta, macrophages. And if we do that cut, what you'll find over several hours, this movie takes place over 4 hours, is those macrophages accumulating at that wound site. Here, I'm going to show you that same sort of a movie, so a cut and then over 4 hours. This zebrafish fin has been just incubated with a solution of a protein that's derived from a different bacterial species, one that seems to, well, I'll just play the movie. Here again, we have macrophages, but they kind of come around here, and then they seem to kind of get confused. A few of them go to the wound, but compared to here, many of them are not migrating all the way there. They're migrating and then kind of getting confused and stopping. So this movie is very recent, just a couple of weeks old, but it suggests that there's ways in which bacteria can influence each other's interaction with the immune system that are very exciting to think about. So all of these things, one might wonder, is this just some peculiarity of zebrafish or peculiarity of these kind of somewhat minimal systems for looking at the gut microbiome? And what I would argue is that, no, it's not. And really what it kind of illustrates is what I think we should be thinking about when we think about gut microbiomes. So these phenomena that we've been seeing all have to do with quite general processes, like intestinal transport. That's what intestines do. That's what your intestines are doing. That's what zebrafish intestines are doing. They transport stuff along. Similarly, bacterial aggregation, whether in the gut or in other contexts, aggregation is something that bacteria do, or transitions between aggregation and motility are things that bacteria do. So we think that these general processes and the kind of general themes that they intersect, that aggregation, motility, and transport are all related, should be relevant to basically any gut microbiome. So these physical processes should be quite general ones to think about. So keep that in mind whenever you're thinking about gut microbes. Maybe just for future conversations and things like that. We also look at various other things, including interactions between bacterial species and a bit of stuff with multi-species communities, and not just one or two species present. Recently, we've been working a bit on trying to make kind of gnotobiotic or kind of controllable ecosystems where we have bacteria, zebrafish, and prey species for zebrafish. So this movie right here is something called a rotifer. It's this little micro animal. The fluorescence specks are bacteria. You can hopefully see little swirly patterns of this guy filter feeding there. So we've been developing techniques to try to control or kind of clamp down on the rotifer microbiome so that we can use rotifers as food for zebrafish while controlling their bacterial state. It's a convoluted way of saying trying to make germ-free rotifers for germ-free zebrafish, which is probably a good Dr. Seuss story that we're also starting to look a bit at kind of larger scale things of zebrafish behavior. And as Dan mentioned back at the beginning, I'm actually quite generally interested in kind of science communication, and especially biophysics in general is just this wonderfully vibrant, exciting field, which I think it's much, whenever people hear about it, I think people are very excited about biophysics, but it's something that doesn't intersect a lot of popular media about science, whether it's physics or biology. So I've been working on things like writing a book, which came out a couple of years ago, which was a lot of fun, and other kind of communication things as well. So I will leave this up, and I'll be happy to take more questions. Thanks.
Thanks, Raghu. Do you have any questions over there?
I don't understand why the aggregation or motility affects the efficiency of expulsion. I imagine expulsion is if it's just pushing away the contents, it doesn't get affected by any motility of the compounds. Is there any like hydrodynamic reasoning or something?
Yeah.
Could you repeat that?
Oh, yeah. So the question is really kind of what's the kind of coupling between kind of the gut motility and the overall transport of stuff. So, I mean, so first of all, if the gut weren't contracting at all, you wouldn't have any transport. And if you are contracting, it's not that you're completely squeezing off the gut and pushing everything forward. You're just kind of, moving the tissue. So imagine you're squeezing some toothpaste, and you're kind of your hand is moving along and squeezing. The more you're squeezing, the more stuff is going to come out. Right? You can try that at home. So it is really that. Not quite sure how. Yeah.
I might misunderstood, but I thought you were showing that the loss of motility
Mhm.
or loss of aggregation affects the rate of expulsion. In your program.
Oh, it's more that, so as you're kind of squeezing these things, the larger aggregates, again, because you're not completely pinching off the gut, the larger the aggregate is, the more it's kind of affected by that squeezing motion. So the very small aggregates don't care so much about that squeezing. The larger the aggregate is, the more likely it is to get pushed out. So that's the connection between the aggregates and the squeezing. So one thing you may be getting at, which is an excellent point, what I just said sounds very kind of qualitative, and it is. There's actually remarkably little kind of actual fluid mechanical kind of calculations for like gut transport, which is actually kind of surprising given how important it is. But I've actually talked to like quite a few people. Like, there's very little like really kind of detailed fluid mechanical studies of this kind of thing. So if anybody's interested in that, you should definitely do it.
Jay.
Yeah, you made a comment that you didn't think this involved signal transduction pathways, but you also pointed out that the macrophages are getting activated.
Right, yeah.
And they're secreting cytokines.
Yeah.
Many different ones.
Yeah.
And so the whole system could respond to that.
You're-
I wonder, like, how do you rule that out? And a related question is, have you ever tried to image cytokines or something like that?
Oh, yeah. So to deal with the second question first, we have not tried to image cytokines or anything like that. Even the kind of just TNF-alpha gene expression, we haven't done much like time result. We haven't done like really the dynamics of that, which I think would be really interesting. The first point, so when I said signal transduction, we don't need to invoke it, that was a little bit of a intentionally provocative point in a way. So it's not that we certainly can't rule out that there are changes in signal transduction. And in fact, everything must be responding to this. There undoubtedly are changes in signaling going on. But my point is just that you could, given cell redistribution, you can imagine a perfectly plausible mechanism that does not require the invocation of changes in signaling pathways. That if you say, okay, let's imagine the known macrophage enteric neuron signaling, like through BMP pathways and stuff like that, is all that's going on. Just taking these two farther apart will have this effect. So it's not that there is not other signaling going on, but it's not necessary to invoke it. And it could well be that there's, and I'm sure especially at longer time scales, there's lots of downstream signaling effects to this. Yeah, it's a great question, and we need to look more directly at a lot of these signals.
Oscar.
You actually didn't mention that these microbiomes have confer any benefits to
Oh, yeah.
zebrafish. Do you know something about that?
Yes, so definitely actually these, they do. And in zebrafish, as has been shown also in like mice and things like that, the germ-free zebrafish are definitely kind of altered in their development compared to ones with microbes. So you see less just like cell proliferation, including in quite specific contexts, like development of the pancreas, like the production of like the insulin-producing beta cells is much less in germ-free fish than in fish with microbes. So, excuse me, things like number of immune cells, number of like pancreatic beta cells, and various other things. Yeah, they're definitely responding to the gut microbes, and yeah, it is helpful to have the microbes there. Yeah.
Does it depend on the types of microbes? Or?
Yeah, well, in many cases, I think that's not well understood, but in some cases, in fact, with like the pancreatic beta cells, it seems to be, this is actually work from our collaborator, Karen Gilliman at Oregon, there seem to be specific secreted bacterial proteins that seem to be the that seem to stimulate like beta cell development and stuff like that. So in a few cases, there seem to be quite specific signals coming from the bacteria. Yeah, which is quite neat.
One last question in the back.
When you added the cholera or the vibrio that your contractions got, there's a lot more heterogeneity
Yes.
in contraction.
Yeah.
Is that because there's a lot of variability in how efficiently these things colonize, or is it a variability in the response?
It's a variability in the response. So these vibrios, the zebrafish native vibrio routinely colonizes to high levels very robustly. So we don't see that much variability in its number. But the fish response seems very variable, and yeah, I don't know. Yeah, we don't know why that is. It's very striking.
One last question in the way back.
I was just wondering about the scaling. I mean, these are so small in the bacteria, obviously don't change in size as you scale up to adult fish or larger organisms. So do you have any thoughts on that?
Right, that's a great question. So what I suspect is that where the scaling might be kind of interesting and kind of transferable to other things is actually kind of the distribution of aggregate sizes. So I didn't show that, but if you make a histogram of the sizes of aggregates, it has a kind of rough power law behavior that is kind of overall set by these kind of kinetic processes and like overall constraints like the gut diameter and that kind of thing. So my suspicion, and we don't know this, is that that kind of scaling would exist in like humans or adult zebrafish and stuff like that, just with different kinds of cutoffs. But we don't know that for certain. It's a great question.
Okay, thanks for a great seminar.
Thanks.